A better 5 a day. How new gene editing technologies like CRISPR can improve vegetable crops for the future. How well a plant grows is determined by complex interactions between the environment, soil, other living organisms, and the plant's genetics. Gene editing can be used to change characteristics of the plant to ensure good outcomes, such as preventing damage from disease. Selective breeding has been used for decades for genetic improvement, producing crops with high yield, disease resistance, or better adaptation to their local environments. But this is slow, taking many generations, and can result in unwanted changes. Traits, such as size, are controlled by genes. Variation within genes can determine different outcomes of a certain trait, such as small versus large. Gene editing traits can be used to make specific changes to DNA in order to change a trait controlled by a gene. Many modern gene editing tools have been developed using similar principles, but the most popular is termed CRISPR. CRISPR gene editing requires 3 key components: the Cas enzyme, the guide sequence, and a repair template. Each of these components enters the plant cell. The guide sequence is a molecule that is designed to match and stick to a target gene that is to be edited. The guide sequence leads the Cas enzyme to the target gene. The guide sequence binds to the DNA and separates the DNA strands. This allows the Cas enzyme to break bonds between the DNA strands. The target gene has now been cut by the Cas enzyme and the complex leaves. The DNA repair template contains any desired changes in the gene. The DNA repair template is used as a copy to enable repair of the break. This results in these precise changes and no other foreign material being incorporated into the edited gene. The edited gene will be expressed, resulting in a new or improved characteristic in the vegetable crop. Gene editing technologies are a faster and more precise method to achieve the same crop improvement goals as traditional breeding. This precision editing technology has been used to improve more than 20 crops, including vegetable crops, such as tomatoes, potatoes, and many others. An example of crop improvement is creating disease-resistant crops. The yellow spots and poor crop yield indicate that this tomato plant has a powdery mildew infection. A small deletion in a key gene was made. This technique means plants can be modified without the addition of foreign DNA. This allows the tomato plant to become resistant to the infection, resulting in an improved crop.